The overall goal of this procedure is to visualize retinal circulation of blood cells in the mouse using a scanning laser ophthalmoscope.
This method can help answer key questions about retinal diseases such as what is the retinal flow dynamics of WBCs and RBCs and how can their flow dynamics contribute to disease pathogenesis.
The main advantages of this technique is that it can be applied on different disease phenotypes and it is also very easy to perform.
This procedure will be demonstrated by our team members Praveen, Bobo, and Neha.
To isolate the mouse blood cells, layer freshly collected whole blood on top of a 1:1 polysucrose and sodium diatrizoate solution in a 2 ml tube. Then centrifuge the tube for 30 minutes. Next, aspirate and discard the plasma supernatant. Then, carefully collect the buffy coat layer composed of leukocytes and the underlying layer of erythrocytes in separate tubes. Next, wash the erythrocytes with 1x PBS to remove any contaminants and resuspend them in 1 volume of PBS. Then, transfer half milliliter aliquots of the suspension into microcentrifuge tubes. Spin down the aliquots, discard the supernatant, and add about 200 microliters of 40% 1x PBS to the pelleted erythrocytes, mix them well, and incubate them at room temperature for 5 minutes. After 5